Hello. First of all, let me thank the organizer, and especially Hari, to invite me to this really beautiful and interesting meeting. Hari asked me to talk about biological insights taught by clinical responses to novel HPV/HDV drugs. And actually, as a molecular biologist, I turned that a little bit around, and I hope you apologize for that, and call it predicted outcomes of clinical responses taught by biological insights into HPV/HDV biology. Finally, we will make a connection, or try to make a connection, between clinical observations that are now coming out more and more in trials and theoretical backgrounds that we have for our understanding of HPV and Delta virus. These are my disclosures. Now, HPV and HDV share the same envelope proteins, follow similar entry pathways. They use the same receptor, and using that receptor, they establish circular episomes, circular RNA or cccDNA in the nucleus of infected hepatocytes. The maintenance of these two molecules, which are not integrates for replication, is crucial for the persistence of these viruses. And they differ. They differ with respect to their behavior during mitosis. HPV cccDNA has been shown to be completely lost during mitosis and has to be probably replenished by de novo entry of virus into cells. In contrast, HDV, that of course uses the same pathway, can spread via cell division in the absence of HPV protein. So a cell that does not have an envelope protein divides can spread HDV. And HDV can assemble and spread using the envelope proteins that are encoded by integrants. I'll come to that in a moment. So there is a possibility of spreading via an extracellular route in the absence of a co-infection with HPV, no cccDNA dependence. Now let's look at the novel targets that interfere with HPV replication, or at least some of them that I want to stress here a little bit. You all know the replication cycle: income of the virus, cccDNA formation, transcription, translation, and we have the encapsulation of pregenomic RNA, reverse transcription, that's addressed by nukes. And then we have the hypothesis, or the finding also of a recycling nucleocapsid that replenishes the cccDNA. Beside that, I want to stress a little bit the fact that beside the double-strand linear DNA, we will also get import of double-strand linear. So beside the relaxed circular DNA, we have double-strand linear DNA that comes into the cell and that integrates readily into the chromosome and encodes proteins. So if you look at the direct acting antivirals, we have entry inhibition, inhibition of capsid transport to the nucleus, but also of capsid assembly. We have inhibition of cccDNA formation, inhibitors of transcription like siRNAs or ASOs, and they are HPX dependent one, and those that are independent of HPX. And we have a group of inhibitors of HPS antigen assembly. In addition, we have a lot of immune modulators, and I will not stress at all these possibilities, but I want to focus a little bit on some basic insights that are related to capsid transport inhibition and inhibitors of transcription, or at least inhibitors that interfere with messenger RNAs of HPV. Now, the three main questions I would like to address is: what are the consequences of the HPV integration on the infection of HPV and HDV? Does integration affect the persistence of cccDNA in an infected hepatocyte? And would cccDNA elimination contribute to HDV clearance in chronic hepatitis delta patients? These are some of the many important questions we have to answer if we go for therapeutic approaches. Let's start with the integration. Thomas Du in my lab worked for some years on in vitro integration. And it was known at that time that 5 to 10% of envelope variants that you find in cell culture supernames, but also in patient, contain double-strand linear and not relaxed circular DNA. And these double-strand linear DNA containing particles can enter hepatocytes via NTCP. This is shown here on the right-hand side. Here we measure integrants, and here we measure the infection. We can block infection and integration by myocludex, telling us that this integration can be blocked by an entry inhibitor. And they instantly integrate. Within days, we have already the first clones that then later on may expand. And these integrated HPV DNA sequences look typically, as in a patient, that they are able to encode for HBs antigen and also for the X protein. And that's a consequence of the nature of integrants. They can propagate via cell division and clonal expansion. And during a natural infection, we can envisage that turnover and replenishment by hepatocytes that carry integrants lead to clonal expansion, and that has been shown, of course, in patients, hepatocellular carcinomas as well, where clonal expansion is a major driver. But how does integration affect HBV and HDV replication? This is a major question that also we ask on the basis of that findings. Now, therefore, we constructed cells that carry artificial integrants. This is one of the cell lines that carry a 2.7 kb HBV fragment and in addition express the receptor. If you use such a cell, compare it to a receptor-carrying NTCP, receptor-carrying HepG2 cell, and infect them with delta virus, you get infected, delta-infected HepG2 cells, and you get, as you would expect, also infected cells that express an integrant. So the integration per se does not interfere with the entry of the delta virus. But one of the surprises came up when we tried to infect these cells with HBV. While the HepG2 NTCP cells can be readily infected, as seen here, by staining HBs antigen, we see a complete exclusion of infection towards HBV. And the same is true if we try to infect the HepG2-15 cell, replicating HBV DNA, expressing the NTCP. There is infection with delta, but no infection with HBV. We have a virus-specific, HBV-specific super exclusion of infection. So cells expressing HBV proteins from integrated HBV DNA support delta infection, but are resistant against HBV infection. And if you imagine a clonal expansion of such integrants in a natural infection, that would mean clinically that the replication space for HBV would be reduced if integrants expand, for example, in e-antigen negative patients. But we would provide additional space for delta that would use the integrants for envelopment. That's a clinical implication of this finding. Now, in my lab asked himself, what protein within the integrant would be important to mediate that infection exclusion of HBV? And we were looking to the envelope protein, and making a long story short, what he found, if he expresses a fragment of the L protein that just contains the capsid binding site, he could see the same effect. So this protein expressed in a hepatocyte excludes HBV from becoming infected. That's the control cell without expressing that, without interfering with delta. And quantification showed that it's almost a full exclusion of HBV infection. Now, intracellular cytoplasmic expression, and he looked even in shorter fragments, expression of the HBV capsid binding site located to the cytosol blocks establishment of an HBV infection. How could we explain that? Our hypothesis and our model at the moment is that the presence of the nucleocapsid binding site at the cytoplasm prevents nuclear import of capsid and redirects both incoming and newly synthesized nucleocapsids into the secretion pathway. So that would mean an HBV particle that could enter a naive cell would go to the nucleus, whereas an HBV particle that enters into a HBL protein expressing cell would be redirected to go out, whereas the HDV particle is not affected by that. The implications would be that cccDNA formation is impaired when an L protein or a truncated L protein is present at the ER. It's a redirection to a secretion pathway, or it's an abrogation of cccDNA formation. De novo formation of cccDNA by reimported relaxed circular DNA that contains the nucleocapsid is probably by the same way impaired after cell division. But this is a hypothesis we have to check. Now, how stable is the de novo synthesized cccDNA? That's of course a very important question, and we addressed that question by a molecular biology approach that I show here. So here's the incoming nucleocapsid going into the nucleus. And of course, it requires the capsid protein to travel to the nucleus. But what happens if we mutate the capsid in that genome that is transported into the cell? That would mean that the de novo synthesized capsids in this infected cell are impaired. There is no de novo synthesized capsids. We can produce such cells if we co-transfect the deficient over genome length genome with a core and complemented with a core protein, and we were surprised that we at all can produce these mutants. So we have a delta core mutant, and we have the wild type, and this is a cesium chloride gradient telling you that that the virus is produced. Now, what happens if we infect? As you can see here, you can infect with that virus a cell. This is wild type infection, S staining. This is delta HBC infection. We can block both infection with nucleodex, telling us it's a proper entry. If we then follow the kinetics of antigen and cccDNA production, we could see over a long time, over 9 weeks, that's a long-term infection experiment, we could see that S antigen kinetic of both viruses is almost the same. E antigen is produced only by wild type, of course not by the core minus mutant, but the cccDNA also remains the same. So during the 9 weeks of infection, the cccDNA levels are the same. Newly synthesized cccDNA molecules are stable for more than 9 weeks post-infection. And in addition to the L protein-mediated restriction of nucleocapsid import, the stability of the ab initio form cccDNA is very high. And if you compare that with recent clinical data that have been provided by Ed Gain at the EASL meeting, and this is just as an example, the long-term study of the combination of Vepicovir and a nuke in patients with e antigen positive and negative chronic HBV, you can see that some of these arms, study design is shown here, receiving more than or about 76 weeks of a combination of nuke and Vepicovir, and then stopping the treatment and looking at possible relapse or sustained virological effects. The disappointing results were that within 4 weeks after stopping the drug, we have 94% of patients relapsing. And the lessons that we here learn is, beside that they could show the author that CAM and nukes are additive in suppression of replication. There was no HBs antigen clearance, no sustained HBV DNA suppression, telling us that the stability of the cccDNA reservoir for more than 1 year is confirmed, and it's in agreement with what I showed you before. However, of course, this is the first generation of a CAM. We will see whether second generation of CAMs improved, for example, by interfering also or more better with the cccDNA de novo establishment. Now let me finish with one aspect of an siRNA treatment. This is an experiment and a publication from Maura's lab, where they used the UPA-SCID transplanted HBV mouse model and reduced all viral markers with an siRNA. And this siRNA is also reducing HBx antigen, as it has been shown here. This is the non-treated control. That's the siRNA, that's X antigen expression. And what you can see is that there is no effect on cccDNA. Now, what happens if RNA treatment is stopped? Then we have a reappearance of the SMC5/6 complex. So here you see in red the viral markers, SMC5/6 in green, and you can see that after 4 weeks of stopping siRNA, we get reappearance of SMC5/6. So the therapeutic application of siRNA or ASOs induce a dual effect. On the first hand side, and that's trivial, they directly target, for example, transcripts like the S antigen. But in a second pathway, they silence the cccDNA-mediated transcription via HBx knockdown, and that leads to SMC5/6 restoration. Now, what happens if we add an entry inhibitor during the follow-up study? Here again is the 4 week of siRNA treatment, the restoration of SMC5/6 protein, and then after follow-up, there is reappearance of the HBE markers and disappearance of the SMC5/6. If we block during the follow-up time entry with bulevirtide or with hepcludex, then we prevent the reappearance of the viral markers and we maintain the SMC5/6 restoration in primary human hepatocytes of these mice. Now, this is an important observation, and the hypothesis is that de novo infection events are needed to re-establish cccDNA activity, and we have to consider these kind of therapeutic combinations in siRNA-treated patients. So the conclusion for me is there is much more to learn about the effect of an entry inhibitor on HPV replication. It's not only preventing the entry of the virion, but probably also regulatory factors. Now let me sum up. Those aspects they are addressed here. I showed that cccDNA persists in vitro in the absence of de novo core protein production. DNA radically integrates into host chromosomes and mediates superinfection exclusion of HPV, but not delta. Expression of the envelope protein restricts the replenishment of cccDNA by preventing nucleocapsid import. Cell division of HPV infected hepatocytes induces cccDNA loss. And the entry inhibition by bulevirtide prevents reactivation of silenced cccDNA, siRNA silenced cccDNA in a mouse model. Now, clinical implication: to achieve a sustained HBs antigen negativation, integrated HPV DNA needs to be therapeutically addressed, probably by cell killing. I think there's a need for immunological restoration. An induction of cell proliferation in combination with suppression of cccDNA may be a key. With that, I would like to thank you for your attention. I would like to thank all my former co-workers and the funding agencies, Ralph Bartenschlager, and my co-workers that are listed here. Thank you very much.